Before we can complete our discussion of the organelles which are found inside eukaryotic cells, we have to consider the two energy processing organelles, which are the chloroplasts and the mitochondria. And these two organelles have something in common, which I think is kind of interesting, that they're both related to energy processing. They both have almost opposite chemical functions, and that they are both formerly, they are descendant from free-living bacteria that used to be their own organisms, but ended up inside of larger cells and forming a symbiotic relationship with those larger cells. They ended up doing some work, finding a compatible friendship with these larger cells, and eventually, through evolutionary time, they lost any kind of features that would have allowed them to be free-living because it was just a better relationship to be part of a whole, and they ended up being part of our cells, or of plant cells in the case of chloroplasts. Now, how do we know that? Well, part of the reason we know that is that chloroplasts and mitochondria both have their own DNA, and that DNA is somewhat more similar to bacterial DNA than it is to the kind of DNA we find inside the nucleus. An interesting consequence of the fact that mitochondria have their own DNA and that they do their own cell division independently of the rest of the cell, when conception occurs in humans, when the sperm meets the egg, only genetic material from the sperm is donated to the egg. All of the mitochondria in that fertilized zygote for that first cell that starts out that organism, that starts out that person, all of the mitochondria come from the mother. So all of your mitochondria are genetically identical to your mother's. You don't have any genetic material inside of your mitochondria from your father. So you can actually trace maternal heritage through mitochondrial DNA and trace history through maternal lines, just like you would be able to trace paternal history using Y chromosome data through patrilineal lines. Really cool research. I did see a paper recently that said that 1 in 100 cases or something like that, some male mitochondria does end up coming in for fertilization, but still really neat stuff. But enough about genetics, that's Unit 5, man. Let's talk about organelles. Let's start out with these chloroplasts. Chloroplasts are the organelle that you find inside leaves that do photosynthesis. So you'll find them in the green parts of plants. Any part of a plant that doesn't do photosynthesis, you're probably not going to find chloroplasts. So you won't find chloroplasts in every plant cell. You won't find them in root cells. You won't find them in bark. You won't find them deep in the wood of a tree. You will find them in the leaves. And if it's an herbaceous plant, you'll find it in the stem as well, green stems. Think dandelion, clover, any other kind of non-woody plant you might interact with. Now, what is photosynthesis? Well, the prefix photo refers to light. So photography is to write with light. Graphy is to write. So photo is light, synthesis is to make, so we're making something using the power of light, photosynthesis. What are we making? We are making sugar. So we're going to take the energy that is in light and we are going to use that to construct sugar, which you may remember from the biological molecules section. Sugars are really good at energy storage. So photosynthesis is the process by which we take energy in light and we convert it into chemical energy for storage. And then we can take the sugars and we can pack them into starch for plant storage polysaccharide. And then when we eat them as animals, we can, take that starch and we can convert it into glucose to burn real quick for our cells in order to give us a boost of energy, or we can convert it over to glycogen to pack in for when we need it later on for ourselves as well. So we need this process as well. We are just as reliant on photosynthesis as plants are, in a slightly more second-hand kind of way. Now, the chemical equation for photosynthesis is pretty straightforward. You take 6 carbon dioxide molecules like we breathe out, right? They're just in the atmosphere all the time. In fact, we're accumulating a little bit too much of them in the atmosphere at the moment, so plants are really doing us a solid by taking them out of the atmosphere. Take 6 of these carbon dioxide molecules and mix them with 6 water molecules, which plants will pull up from their roots, or if they're an aquatic plant, they'll just take them right from directly around themselves. So 6 carbon dioxides and 6 waters, and you mix them together and you get yourself C6H12 CH12O6. That should sound familiar. That is the chemical structure of glucose. It's also the chemical structure of fructose and galactose and any other hexose sugar. C6H12O6 is any 6-carbon sugar that you like. So they've made sugar here. Hi, as I'm editing this in the future, I'm realizing that the oversimplified chemical equation that I'm presenting with here might be a little bit misleading, because if you just mix carbon dioxide and water, you will not get glucose, you'll get carbonic acid, regardless of how much sunlight you flood your system with. So the trick of photosynthesis is more than just catalyzing this chemical reaction. It has to be a good deal more clever in order to enable the capture of that radiant energy in the form of chemical energy, which is why we're going to spend such a long period of time in Unit 3 talking about just how clever chloroplasts really are. But there's a problem with that. If you have 6 carbons on the left-hand side, okay, and we got 6 carbons on the right-hand side, that works out just fine, so we got that. And we have 6 times 2, 12 hydrogens on the left-hand side and 12 hydrogens on the right-hand side. That works out okay. And we have 6 times 1, okay, so 6 oxygens, plus 6 times 2, so there's 18 oxygens here on the left-hand side. And we have 6 oxygens in the sugar. And we have 12, so we have too many oxygens. What are we going to do? We need to pair them off together in order to make this equation make sense. So a waste product of photosynthesis is oxygen production. Plants don't need the oxygen, at least not right away. They're going to need it if they want to break down the sugar later on, but they don't need it right away. So they just let that oxygen float out into the atmosphere. Every molecule of oxygen that you have ever breathed in has essentially come from this process. Maybe not by plants. Some of it's going to be done by bacteria. Some of it's going to be done by protists. But every molecule of oxygen in our atmosphere has essentially come from photosynthesis. So thank a plant next time you take yourself a nice deep breath in. Now, bacteria can do photosynthesis, plants can do photosynthesis, and protists can do photosynthesis. Fungi do not do photosynthesis, and animals do not do photosynthesis. But chloroplasts themselves are a membrane-bound organelle, and prokaryotes like bacteria cannot have membrane-bound organelles. They're too small and simple to have that kind of thing. So bacteria, when they do photosynthesis, they have to use their own cell membrane in order to get the process done. So only plants and protists, eukaryotic cells, can have chloroplasts inside of them. Bacteria never can, okay? So chloroplasts are special for plants and certain types of protists. Now you're thinking, I want a little bit more detail than that. That was basic. I know C6H12O6. I know carbon dioxide and water is how you make sugar, and you rearrange things and you store the energy of sunlight into chemical energy. Give me a little bit more than that. All right. Here's the structure of a chloroplast right here. It's actually got 2 membranes around the outside. So it's not just a membrane-bound organelle, it's a double membrane-bound organelle. And then if you look at the interior, there's even more membranes on the inside. It's made up of these stacks of membranes, these little green pancakes on the inside. I think of them as pancakes. They're called thylakoids. That's not something you need to know this unit. You don't need to know the structure in that much detail. Unit 3, though, you will. So the next unit that we study, you'll need to know the structure of a chloroplast. And these thylakoids is where the light harvesting machinery is located. This is where we find pigment molecules. What's a pigment? A pigment is a chemical that absorbs particular wavelengths of light. So when light hits an opaque object, a non-transparent object, some of that energy from the light can be absorbed. It can be retained. Imagine when you go outside and it's a very sunny day outside and you're wearing like a black t-shirt, it's going to feel really, really hot, and much hotter than if you were wearing a white t-shirt. Well, white t-shirts reflect light. It all just kind of bounces off you, whereas the pigment in that dark t-shirt, that's going to absorb more of that energy, and that absorbed energy is being re-radiated out as heat. But in the thylakoids, when they absorb that light energy, they're able to capture it and use it to secure it into chemical energy instead. So the same kind of idea is at play here. And the pigments inside these thylakoids, inside the chloroplast here, are really good at capturing just about every wavelength of visible light except for one. The one wavelength that plants are not very good at utilizing for photosynthesis is green wavelength. If you were to ask a plant what its least favorite color is, it would probably tell you green. And it's just a kind of an interesting quirk of evolutionary history. Why are plants green, right? Why is that the one color they're not good at utilizing? And there's a lot of thoughts on that. There's a lot of speculation on this idea. Here's one thing we do know. If they were able to absorb all of the wavelengths of light, it would kind of be like that dark colored t-shirt. It would just get really, really hot in the leaf, and that might end up denaturing some of the enzymes and proteins involved in the photosynthetic pathways, and that might make photosynthesis less efficient. So it's probably true that some of the light needs to be excluded. Some of it needs to be reflected off, but for whatever reason, green light is the one that's reflected off, and there's a lot of papers with speculation about why that might be. Now, there's a lot of pigments, and we'll talk about, I think, 4 or 5 of them in Unit 3, but here's 2 of the big ones. Chlorophyll A, which absorbs mostly blue, violet, and red light, and it participates in the first stage of photosynthesis, which we call the light reactions, because this is the part where sunlight is directly involved, or light is directly involved. And then chlorophyll B absorbs mostly blue and orange light and participates indirectly in the light reactions. So notice these 2 main pigments, they absorb blue, they absorb violet, they absorb red, they absorb orange. They don't absorb green though, so green is reflected off. Consequently, when we look at leaves, what do we see? We see green light. It's the only thing bouncing off of them, right? So that's why we see leaves as being green, and that's true of all color. That's what color is. It's the light that's reflecting off of the object. Now you say, I want even more detail. Well, wait for Unit 3. We're going to take a deep dive into photosynthesis and the structure of chloroplasts, and you will know more about photosynthesis than you could ever possibly have wanted to know. And if you still want more after that, I have some recommendations of which class to take next and which professors to take it from, but hopefully I'll get you a good start. So I mentioned that photosynthesis is part of energy processing, which puts it into this category of chemical reactions called metabolism. Metabolism is the chemical processes that occur within living organisms in order to maintain life. Photosynthesis is a particular type of metabolism which we call anabolism. Anabolism. You've heard of anabolism before in a different context. Anabolic steroids, right? They help build up muscles. Anabolic reactions build things up. So in the case of photosynthesis, we're building up sugar molecules. We're taking tiny little carbon dioxide molecules, rearranging them to make glucose molecules, much larger molecules as a result. And in the process, we are storing light energy. Typically, anabolic reactions store energy as they go. And you can think of that as charging up a battery. And as a nice bonus, we get oxygen gas. Now, the exact opposite reaction is something we call cellular respiration. This is where we use oxygen gas in order to break down glucose. We say we oxidize glucose in order to release that chemical energy and make our cells do work in the process. And that is the domain of mitochondria. Chloroplasts may only show up in certain kinds of plant cells and certain kinds of protist cells, but mitochondria show up in every kind of eukaryotic cell. There are some narrow exceptions, but they are basically as iconic to eukaryotic cells as the nucleus is. They are fundamental to eukaryotic cells. They make energy processing so efficient that they basically enable large, complicated eukaryotic cells to exist in the first place. Mitochondria are where it is at. They are fascinating structures, so let's talk about them a little bit. They are involved in a process called cellular respiration. More specifically, they are responsible for all of the processes involved in aerobic cellular respiration, all of the oxygen requiring portions of cellular respiration. The glycolysis stuff, the anaerobic stuff, fermentation, that can happen outside of the mitochondria. All the stuff that requires oxygen, though, that's in the mitochondria. Photosynthesis was the process by which we took energy which was in light and we stored it into chemical energy. Cellular respiration, on the other hand, is where we're going to take the chemical energy inside sugars and we're going to turn it into something more useful. We're going to turn it into the chemical energy inside of another kind of molecule called ATP, adenosine triphosphate. Adenosine triphosphate is a modified nucleotide. It's like DNA. It's actually like RNA. But with some extra phosphate groups on it. It's called the energy currency of the cell. It's like an energy token inside the cell. You can go to any protein inside the cell and spend some ATP and you can get that protein to do its job one time. Go to an enzyme, put in an ATP token, you get it to do its job one time. Go to a muscle fiber, put in ATP, you get it to contract one time. Go to a transport protein in the cell membrane, put in an ATP token, you get it to let in a molecule one time, right? So ATP is this energy currency of the cell. It's what the cell uses to enable its enzymes and its proteins to do their work. So cellular respiration is the ATP making function of the cell, and the mitochondria are what gets a lot of that ATP produced. So that's why they're referred to as the powerhouse of the cell. A phrase which I'm sure is already cemented into some of your brains, but is a phrase which does not appear on even a single one of my exams, so that is not something you should spend your time memorizing. It might be helpful as a kind of mnemonic for kind of remembering what the mitochondria does, but what you want to remember is that the mitochondria is involved with aerobic cellular respiration, which is the process by which the cell is going to use oxygen to break down sugars and produce ATP. And in so doing, about 40% of the energy in our food can be converted into energy of useful work, such as the contraction of muscles from moving around and swinging a hammer and raising a barn or whatever other kind of work you have to do. You can see on this slide that the mitochondria has a couple of membranes, just like the chloroplast had more than one membrane. It's got an outer membrane and it has an inner membrane. The outer membrane is kind of kidney bean shaped. It defines this outer border, this outer shape of the mitochondria here, and it's not overly complicated in its structure. Meanwhile, the inner membrane is very, very complicated in its structure. It folds back and forth and back and forth and back and forth, and that's to increase surface area. Remember that surface area thing we talked about right at the beginning of Unit 2? And the reason is this membrane has a bunch of enzymes on it. We're going to talk about something called an electron transport chain, and all of those electron transport chain enzymes are on this membrane. So the more surface area there is on this membrane, the more work this mitochondria can get done. So it behooves it to have as much surface area as possible on this membrane. Now, if you're not clear on how folding increases the surface area of a membrane, imagine it like this. Imagine you have a really big bag and a really small bag, right? The really big bag has more surface area than the really small bag. That should be pretty easy to understand, that there's just more material that was involved in constructing the really large bag than the really small bag. Now, let's say that you want to put the really large bag inside of the really small bag. The only way you could do it is to fold the really large bag, to crumple it up, right? To twist it and try to crush it down as far as you could. And in so doing, you're going to create a lot of folds. You're going to create exactly what you have here inside this mitochondria. It's going to fold back and forth and wrinkle in on itself as you're trying to crush that and smash that really large bag into that really small bag. So this is what you get. The surface area of the interior membrane is larger than the surface area of the exterior membrane, and the reason is because the interior membrane has those important enzymes and they need lots and lots of space to do their work. Now, just like with the chloroplasts, we are going to talk in detail about how mitochondria work and the process of cellular respiration in Unit 3. So for right now, I'm just going to give you an overview. This is a glucose molecule. Appreciate my high quality animation skills here. We have our 6 carbons, our 6 oxygens, our 12 hydrogens. And before it can enter the mitochondrion is singular, it has to go through a process called glycolysis. Lysis, look at that, lysis again. We're gonna break sugar, sugar, to break sugar. That happens outside of the mitochondrion. We're gonna break it in half, and that actually generates 2 ATP by itself, 2 net ATP. Actually, I have to invest 2 and you get 4, but we'll say it generates 2 ATP. And you get 2 what we call pyruvate molecules. Pyruvic acid comes out of this. These are now allowed to enter the mitochondrion along with some oxygen molecules because, of course, the mitochondrion is involved with aerobic cellular respiration. So this is where all the oxygen requiring processes happen. And that produces a whole bunch of carbon dioxide and water as a byproduct, but also a lot more ATP, 38 more ATP. So the mitochondrion is a far more efficient producer of ATP than any anaerobic process, any non-oxygen requiring process would be by itself. And that's why mitochondria are so important to eukaryotic cells, because without them, you just can't get the amount of energy required for these big, complicated cells. So this is what we're going to talk about largely in Unit 3, is photosynthesis and cellular respiration, and how we can get this much efficiency out of it. Now that we've talked about membranes and transport across membranes, and we've talked about the size of cells, and we've talked about every organelle and what differentiates different cells from each other, I want to draw your attention to the list of the characteristics of life that we talked about in Unit 1, and consider a single cell. Like a single cell, either a bacteria or a cell taken from your cheek, right? A cell taken from a leaf, something like that. Is it orderly? What have we just been studying, right? They have an orderly structure. They have all of these structures that have specific names and specific functions, and we know exactly what they look like and what they do and how they work with each other and how they exchange materials, right? So they're very, very orderly, as a matter of fact. Do they regulate? Oh my God, we spent like 4 videos just talking about how membranes operate in regulation, and how we talked about energy processing functions and how they regulate. Do they reproduce? We're going to talk about that in a moment here with mitosis, but you guys probably know at this point that cells reproduce, that's how they duplicate themselves. Do they respond to the environment? You guys have seen some videos already of some cells swimming around and doing phagocytosis, right? They respond to external stimuli. If you have chemicals interacting with receptors, that triggers them to behave in certain ways, so they respond to stimuli. Do they utilize energy? We just talked about photosynthesis and cellular respiration, so they certainly utilize energy. Do they grow and develop? Yeah, they absolutely do. We'll talk a little bit more about that. Do they evolve? Well, yeah, they do, actually. You have situations like antibiotic-resistant bacteria that emerge. That is an evolutionary process. Those didn't exist before we started overusing antibiotics, but now they're a major problem. And they occurred because we were essentially selecting for those strains of bacteria that already had this resistance genes in them. And since those were the only ones that weren't being killed off, that became the dominant strain. So yeah, individual cell lines can evolve by themselves as well. So cells have all of these characteristics of life in and of themselves. So they themselves are living things.